All right, so as we talked about yesterday, guys, as I put in the tweet yesterday, you do not need to give me the function, okay, of the structures that I am going to circle. You will only have to identify the structures that I circle. There will be 8 that you will have to give me the names of. The 9th question on the quiz is what kind of cell this is. You just write down what kind of cell it is, and that is the 9th question. That's a pretty easy question, I would think. All right, so I would like you to do number 1. And number 3, keeping in mind that number 3 is pointing at the yellow stuff covered with the blue dots, not the blue dots. Everybody follow me? All right, so you're looking at number 3 is the yellow stuff covered in the blue dots, not the blue dots themselves, in case there's any confusion there. Number 5, I would also like number 6, 7, 9, what am I at here? 1, 2, 3, 4, 5, 6. 7, and 8. All right, so I want all of those identified. And then your last question is, what kind of cell is this? Questions? Only the circled ones. All right. Structure number 1. What is it? Cell wall. Okay, for structure number 3, the yellow stuff with the blue dots is the rough ER. If they wrote rough endoplasmic reticulum, good for them. Okay. If they wrote number 5 as being the water vacuole, they are right. Okay, number 6, what is that? Mitochondria. Okay, number 7, the green stuff. That's not the right answer. Green stuff is not the right answer. Okay, it is a chloroplast. All right, that's the photosynthetic organ of the plant cell. So you can mark the last question. It's a plant cell, as if there was any doubt, being that the whole thing is green. All right. For number 9, anytime you got an arrow pointing at what seems to be nothing, it's pointing at cytoplasm. Okay, so we got cytoplasm. Number 10, the layer just inside the cell wall, cell membrane. And lastly, number 12 is pointing at the nucleolus. Make sure they have 2 L's. Otherwise, it is nucleus, and it is not right. Okay, so nucleolus. And again, last question, the answer should be that this is a plant cell. Please give them a mark out of 9. Show it to them, put it on top of your microscopy labs in the box, and then hustle back to your desk. All right, so what we got to go over here, guys, is the cell anatomy lab. All right, you just handed in the microscopy lab, and I wasn't too worried about whether you labeled things on those diagrams correctly or not, but I'm going to be a lot more worried about it, okay, on this lab. Now, we're going to be looking at 4 different things, 4 different cells. All right. Our purpose in doing that is to investigate this problem. Cells have to carry out the basic functions of an organism in order to survive. Thus, they must have structures within their cells that carry out these basic functions. So, when we're dealing with our hypothesis, okay, the if part probably needs to talk about what cells do. Okay. The and part is, as always, a description of the experiment. We're looking at some cells. Okay, obviously don't be that simplistic, but around that idea. All right. The then part needs to tell us that if they got this job to do, what do they all need to have? So the then part should be telling me what you think the cells need in order to carry out those basic functions and be specific. Okay. All right, so talk about the structures. Now, some stuff about the variables. We are going to be looking at 4 different things, 4 different kinds of cells. Well, not kinds of cells, 4 different cells. All right, we're going to look first at what's called on the slides monocot dicot cross section, which is a fancy way of saying someone laid a leaf flat and cut it this way. All right, so that you're looking at the leaf head on. All right, everyone follow me there? Okay, and I'll show you what a picture of that looks like in just a minute, but for you auditory people, okay, what you're going to see there are essentially very long rectangular plant cells kind of on the top, and then you'll see some less rigidly shaped plant cells underneath that and things like that. You're going to be wanting to draw the rectangular shaped plant cells. And in them, you will very clearly see a lot of organelles. Right? You will very clearly see a nucleus. You will very clearly see several chloroplasts. You will see that the chloroplasts and the nucleus tend to be pushed against the outside of the cell, and the cell looks kind of blank in the middle because of the water vacuole. All right, things like that. You gotta use your imagination a little bit. I'm not saying make stuff up, okay, but you do have to look at it and go, well, that could probably be this. If you think it is, then label it that way. You're probably right. All right, but at the same time, again, if you think it's a ribosome, you're wrong. Okay, you're not seeing any ribosomes, but you will see rough ER in a lot of the cells. Right? You will see Golgi in many of the cells if you get them focused well enough. Right? So make sure you're looking carefully at the things that we're viewing under the microscope. Get them well focused, right? And really pay attention when I show you the actual pictures so that you know what you're looking for. No, it just appears dark enough because it's covered in ribosomes that so it's dark enough that it shows up. Yeah, you're not going to see specific dots at all. No. All right. In fact, it'll be hard to recognize as rough ER, except that we've said it often looks like the goatee on the nucleus. Okay, so all right, so that's one of the things you'll be looking at. Okay, is the plant cell. We're also going to be looking at a generalized animal cell, which is actually a piece of what's called stratified squamous epithelium. It's a fancy way of saying that red skin underneath your skin that you see if you scrape yourself real bad. All right, that's stratified squamous epithelium. When you look at that, okay, the cells are a lot smaller. The plant cells are big compared to the animal cells, and quite obvious because of their rigid shape. The animal cells are obviously not rigidly shaped. In fact, when you get it focused, the stratified squamous epithelium will look like mud that has dried. All right, so it's got cracks in it. Okay, you've all seen that happen, right? When mud dries, it tends to crack kind of randomly all over the place. That's what this stuff will look like under the microscope. And you'll see purple dots. Those purple dots are nuclei. So they're a nucleus of a cell. So find one of those and then get it focused, and you'll be able to see the kind of crack in the mud that is the cell membrane, all right, around it. And then you'll be able to start figuring out what some of the blotches inside of the cell are. And this morning, I had in both classes several groups who had theirs focused well enough to see Golgi, mitochondria, ER, rough ER, okay, nucleus and nucleolus, nuclear membranes, okay, like these are, if you get them focused and are patient, you will see these structures quite clearly. For the most part, yes, but there is one that I want to talk about in just a minute. Okay. For the next one is the human blood smear. Now, the human blood smear is we're looking at just to draw a lab diagram. It is not going to be included in your hypothesis, your analysis, or anything like that, because people just always ask, what does blood look like? Can we look at blood? So you can look at blood. All right, but blood cells are different than all the other cells we're going to look at. Red blood cells don't have nuclei. All right, they don't have a nucleus. They also don't divide. Red blood cells are produced by the division of special cells within your bone marrow. Okay, so they do come from the division of existing cells, just not from red blood cells. And so we have to, when you look at them, their membrane is different, because what does a red blood cell do? What does it carry? Oxygen. So its membrane is a little bit thicker. It's laced with hemoglobin, and it catches the stain in such a way that you can't see into the cell. All right, so you wouldn't be able to see anything inside the cell, and there's no nucleus in them because they don't have one. So all you're really going to see is like the cell membrane and maybe cytoplasm, and that's it. All right. So again, they're not going to be included when you're answering the analysis question on this lab. You're going to ignore the fact that you looked at the red blood cell and saw nothing inside it. Does everyone follow me there? Otherwise, it really screws up the analysis, because it's like, whoa, we saw this in everything but the red blood cell, and things don't work out as well. You just have to stain them differently than these ones are stained. Yeah. All right. Then the last thing is you're going to make a wet mount of your own cells. Okay, you're going to go CSI, take a cotton swab, and swab the inside of your cheek. Now, proper technique for this goes as follows, Colleen. Okay, you will twist. Okay, if this marker here is the Q-tip, you twist it back and forth in your finger while you are swabbing the inside of your cheek. All right, so that you get the slime everywhere. All right, and then once it's good and gooey, you wipe it, okay, with the same twisting motion onto the blank slide you used to make the letter E's the other day. All right, so you'll smear it on there real good. You won't see very much, okay, but believe me, you get a lot of cells this way. That's why CSI swabs the inside of people's mouths for DNA tests. The cells inside of there come off real easy. All right, so you'll do that. You'll smear it on, add a drop of water, throw the cover slip over top, and view it the same way you did the last time for the letter E. Now, here's the thing about the cheek cells. They're not stained, so they are not going to be colored like the rest of the cells you looked at. So it's a little more difficult to get them focused because they're transparent. The way to get them focused and make them stand out is to alter the kind of light that is coming in. So underneath the stage was that condenser diaphragm, that tab I showed you the last time. Okay, you want to close that off so that it gets kind of dark. And as it gets darker, the cells will simply jump out. All right, not literally jump off the slide, but they'll jump out appearance-wise. They'll look almost three-dimensional, and they'll be very, very clear. Now, if you see something big and dark on your slide, it isn't a cell. It's something you ate. Okay, especially since it's right after lunch, and probably most of you didn't get a chance to brush your teeth. All right, so you're probably going to have some food detritus coming off on there, and if it's big and dark, it's not one of your cells. Your cells will be transparent on the slide. Okay, now I'm going to show you what these things look like in just a minute. Okay, your variables. Okay, so I did your design part here. Manipulated variable. Okay, what are we looking at? Different kind. Yeah, we're looking at different kinds of cells. It's important if we're going to compare them all, wouldn't you say? Okay, and the responding variable is what we're looking for in all of them. That part I will leave to you. Okay, the controlled variables, as Michael said, a lot of them can be the same as your microscopy lab because we're using the microscopes in the same way. But there's one more controlled variable that needs to be added. All right. What kind of cells did we say we had in our body? Not animal. We had talked about two types of cells yesterday. Close. Eukaryotic. Yeah, eukaryotic cells and prokaryotic cells. We have eukaryotic cells. We're only looking at eukaryotic cells today because prokaryotic cells don't have Golgi and nuclei and stuff like that in them, so it would serve no purpose to look at them. So one of your controlled variables should be that we are only looking at eukaryotic cells, all right? Because that would interfere with the lab if we weren't. All right, when we get down to the analysis portion here, okay, when we get down to the analysis portion, it says, based on your knowledge of cell structure, so looking at the notes from yesterday, the diagrams that we labeled in class, okay, and your observations of different cells in this lab, again, excluding the red blood cell, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? So obviously, between plant and animal cells, there are some differences, and that's kind of part of this, but there's also a lot of similarities, and that needs to be the bulk of what you discuss here, right? It's going to be kind of like a short paragraph type of answer to answer this question, right? What you need to focus on are what things did you see in the plant cell, the cheek cell, and the animal cell? What things were the same? Okay, if they were the same, was there a reason they all had them? That's the kind of stuff you need to talk about in the analysis part of this lab. Does everyone understand? Okay. Now, the diagrams here, the pictures. Okay, so one of the things you're going to be looking at. First, this is here where I've got top and bottom labeled. This is the Dicot leaf, okay, cross section. These are the cells here near the top that you're going to want to draw. Right? They're called Palisade layer cells, and they have nice fixed shapes and makes them easy to see. Okay, if we blow that up a little bit, all right, you can clearly see the nucleus. Your nucleus may not be stained red in yours. In some of the slides, it's stained red, and some of them it's kind of grayish green. All right, but it's a big dot in the middle. It'll be obvious. Okay, you can also see quite clearly all the chloroplasts around the edges of the cells. Okay, and you can see that some of the cells, okay, like this one here, it's clear in the middle. Okay, this one here's got kind of a clear section in one spot. Again, things like water vacuoles and stuff like that. Right? So that's what you're looking for. You get one of those focused, and you'll be able to see quite a few things. All right. This diagram, this picture obviously is kind of blurry, so it doesn't work quite as well as what you'll see under the microscope. Okay. This here, the pink one here with the circle on it, this is stratified squamous epithelium. Okay. The thing I have circled there is a single animal cell. Okay. The nucleus is quite large, and obviously in the middle. But when you look at it, it'll look a little bit bigger. Okay, through your microscope, and you'll be able to see quite a few kind of dyed, stained structures within the cell, and then it's kind of your best opinion as to what is that structure. All right, they're not going to look much like what's in your cell diagram, but do your best to compare. If you think that this thing is a Golgi as opposed to you know, a rough ER, that's your prerogative. All right, realistically, I'm not going to know one way or the other. I'm just going to look at your lab drawing and go, well, I can see how they might think that, and probably give you the marks. Okay. Now, if you draw a big circle in the middle and label it as ribosome, no, you're not going to get marks for that. Okay. It's the nucleus, silly. Okay. All right. Their blood looks like this. Okay. So the bottom left here is the blood. The pinkish orange ones are all red blood cells. These funny looking ones here are leukocytes. They're white blood cells. Okay. And this is what your cheek cells are going to look like, bottom right here. All right, each one of these circled things is a cheek cell. All right, you can see that they are kind of transparent, and this looks quite a bit dimmer than the other two. That's because we took this picture by closing up the iris so that they would actually stand out a bit. Otherwise, it's so bright the light just goes right through them and you can't see them. It overwhelms you. Okay, you guys follow me there? All right, so those are the 4 things that you're going to be looking at in the lab today. Are there any questions on that? All right, so we have got about 50 minutes, okay, to get all of these things viewed, get your pictures snapped of them, and then the lab report will be next week. I'll figure out when here in a minute.